Okay, thank you very much. We will begin now. The title of my lecture is Oxylipins, a cry for help. And the slogan of this meeting, of this lecture, is Oxylipins are more interesting than you think. During the course of this lecture, I will try to explain why oxylipins are more interesting than we think, and why oxylipins are so interesting, and how did I study this group of chemicals. I think all of you could imagine polyunsaturated fatty acids. They are fatty acids with a lot of unsaturated double bonds. And when we oxygenate these polyunsaturated fatty acids, we have oxylipins, oxygenated derivatives of polyunsaturated fatty acids. Here you can see some representatives of oxylipins from different kingdoms of life, from animals and from plants. And it was a short introduction. And then let's take a spotlight on oxylipins in animals, specifically in vertebrates, and in ourselves, in humans. Maybe not all of you have heard about oxylipins, but the word eicosanoid will be more familiar for you. Eicosanoids are just vertebrate oxylipins. They are named eicosanoids because they have 20 carbon atoms in the molecule, and the word eikosi means 20 in ancient Greek. So eicosanoids are just vertebrate oxylipins. Vertebrate oxylipins, including human oxylipins, are synthesized in two pathways: cyclooxygenase pathway and lipoxygenase pathway. The cyclooxygenase enzyme starting the cyclooxygenase pathway is the most familiar for all of us because every person has sometimes taken aspirin, diclofenac, florbiprofen, ibuprofen, and these medications are just biochemical blockers of cyclooxygenase enzyme. Cyclooxygenase pathway results in the production of prostaglandins, cyclic oxylipins, but there is also a lipoxygenase pathway, starting with lipoxygenase enzyme. Lipoxygenase converts arachidonic acid to hydroperoxides. They are further converted to leukotrienes and lipoxienes. This pathway is also familiar for a lot of biologists and for medical specialists. And these two pathways participate in the pathogenesis and orchestrating of inflammatory reactions. Inflammation is an universal response of our body to injury or to a pathogen attack. When a bacterium invades our tissues or something injures our tissues, the cells in the area of injury are damaged, and they start to form oxylipins and release them into the intercellular space. These oxylipins are prostaglandins and leukotrienes, and they are a cry for help from our cells. In the chemical language, they mean, help me, someone is eating me or injuring me. But there are different oxylipins with anti-inflammatory properties. For example, lipoxienes. They are formed from fatty acid hydroperoxides released from injured cells by our leukocytes and dampen the inflammation to counterbalance this reaction and to avoid excessive inflammation. We know that some other chemicals participate in the inflammatory reaction. For example, biochemic amines that are released from the basophils, from mast cells, and cytokines. A lot of protein chemicals, a lot of protein mediators that form a complicated network and enable our leukocytes to communicate between themselves and to coordinate their actions to kill the bacteria and to heal the injury. So our oxylipins are crying for help from our cells that kickstart the inflammation. It is a familiar facts about oxylipins, and maybe it was not worth to start a lecture if I would tell only these facts. But oxylipins are more interesting than you think. And plants can use oxylipins too. Plant pathway for forming oxylipins is different from our one, but please see at this enzyme. It is lipoxygenase. Lipoxygenase, this lipoxygenase is a homologue of our lipoxygenase that forms leukotrienes. This enzyme is a relative of our lipoxygenase, and it performs the same biochemical function. It converts linolenic acid, the prevalent fatty acid in plants, to hydroperoxide. But the further biochemical steps are different. Plants have no cyclooxygenase, but they could also form cyclic oxylipins by the pathway of 3 enzymatic stages: alenoxide synthase enzyme, alenoxide cyclase, and beta oxidation enzymes. It results in the synthesis of jasmonades. They are potent plant hormones. The second branch of plant oxylipin metabolism is hydroperoxidase pathway. It is quite dissimilar from our oxylipin pathways. We cannot split our oxylipins in the middle to make volatile components. All our oxylipins are non-volatile, but it is not the case of plants. Plants split this hydroperoxide and form volatile alcohols and aldehydes that belong to a large group of green leaf volatiles. Let's start with jasmonades. They were first isolated in 1962 and became their name from jasmine essential oil. They were isolated from this essential oil. Jasmonades are potent plant hormones, but the first function that we will consider tonight is very similar to functions of our oxylipins. Jasmonades coordinate plant responses on pathogen attack and on injury, as well as our oxylipins. Plants have no inflammation because they are sessile and their cells are sessile. They cannot move to the site of injury and kill the bacteria that invade the plant tissue. But plant solves this problem in other way. When a bacterium invades plant tissues, the injured cells start crying for help, as well as our cells. They cry for help with oxylipins, for example, with jasmonades. Jasmonates are released and reach adjacent plant cells that synthesize a wide variety of chemical compounds that kill bacteria. These compounds are named phytoalexins, and some of them are widely used in traditional medicine. For example, allicin, the active component of garlic. But jasmonates are used by plants not only to respond to bacterial attack, but, for example, to respond to herbivory attack. In order to understand what could the plant feel when a herbivore attack it, please imagine that you are in a summer forest. In a warm summer forest, in a warm summer day, and this forest is full of insects, mosquitoes, bumblebees, caterpillars, and then it will be a nightmare. You cannot move forever. And these insects are attacking you because you are tasty. It is we have a good news. It will never happen with us, but plants live in this condition all their life, and we can understand the challenge that they face. And they solve this problem in a similar way. When a herbivore attacks plant tissues and feeds on this plant, plant cells react on injury and release jasmonates. These jasmonates reach adjacent cells, and in addition to phytoalexins, they start synthesizing plant toxins. The set of these toxins are different depending on the specific plant species. But I have drawn only the most familiar for us: nicotine and caffeine. From the evolutionary point of view, these compounds are just plant toxins. Yes, we use some plant-derived components as parapharmaceuticals, as food, as beverages, as food additives, as medicines, but plants synthesize them not for us. They are evolutionarily intended for insects and for other herbivores. For example, caffeine is a potent antioxidant acting on caterpillars, for other insects, and the nicotine is also plant anti-insect toxin. But here I can recommend a magnificent book by David Kennedy, Plants and the Human Brain, where these moments and these facts are described better and more detailed. But here I will notice that all of these components are synthesized in response to injury and to release of jasmonates and other oxylipins. But the functions of jasmonates are not restricted to the reaction on injury or to herbivore attack or to a pathogen attack. They regulate a wide variety of stress responses. For example, drought stress, salinity stress, thermal stress, freezing stress, and even leaf senescence. It is a beautiful process that we can observe, that we can see each autumn. And here in my photo art, I tried to capture this process. It is one of the last photo arts of mine. It is named The Last Chlorophyll of an Autumn. This process is coordinated by a wide variety of plant hormones, not only jasmonates. by Pseudomonas also contribute to this process. When the next autumn came, when the next autumn comes, and you see the green, yellow, orange, and red colors of this season, just remember that it is the action of Pseudomonas, the plant oxylipins, that are similar to our prostaglandins. It could seem unusual that plant oxylipins perform such a wide variety of functions. Our oxylipins are just inflammation mediators, and they are not involved in stress reactions. But plants are sessile, and from this point of view, it is the best solution to use injury mediators for stress responses. Fight or flight reactions we are accustomed to are unavailable for plants, and they protect as they can. They use Pseudomonas and other oxylipins to coordinate their biochemical reactions. It is confirmed by a striking similarity between plants and other group of organisms that are evolutionary close to us, but they are also sessile, in contrast to us. I mean corals. They are sessile organisms like plants, and they also use oxylipins to regulate stress responses. We have only scarce data on oxylipin functions in corals, but we know that in corals, oxylipins trigger response to a mechanical injury, like in plants and like in our organisms, and response to a thermal stress. It is dissimilar from our organisms, but very similar to plant organisms. And moreover, the coral lipoxygenase alanoxide synthase pathway is very similar to the plant oxylipin biosynthesis pathway. But we know a little facts about oxylipin biosynthesis in corals, and it is just to be elucidated better. Let's discuss the next question. Can plants communicate to one another? Here is one more photo art of mine. It is named "Wake Up, My Darling." This flower is in love with this flower, and this one tries to warm this one by its light and warmth. But unfortunately, it is only a beautiful fairy tale. Plants cannot laugh, but they cannot. But they can communicate to each other. They have no sound-based communication available for us, but they have another communication. Their Pseudomonas have volatile forms. For example, methyl Pseudomonate is a volatile compound that can transmit by air from one plant to another. And when a caterpillar, other insect, or other animal attacks one plant, it alerts its conspecifics by releasing Pseudomonas. These volatile Pseudomonas elicit the same defense reactions in the adjacent conspecifics. And when this caterpillar reaches this bush, this plant, that it will die because this plant is already prepared to meet it. it has already synthesized a lot of plant toxins. For example, caffeine, nicotine, or other toxins depending on the specific species of plant. But plant oxylipins are more interesting than you think. And here I would like to remind about one fragment of the book Harry Potter and the Half-Blood Prince. Here, one portion is discussed. The most potent love potion that is called amortentia. And this love potion smells like a fresh mown grass for Hermione Granger. I know that in the book it is just magic, but this effect could be reached without any magic if the potions master adds some plant hexanols or hexanols, volatile plant oxylipins, to the cauldron. These plant oxylipins are the compounds that are the cause of the smell of freshly mown grass. And I think that this love potion may contain these compounds if we could discuss this potion from a biochemical point of view. And it could fit into the biochemical logic because amortentia is a love potion and it needs to contain some food additives that enhances its smell and taste. Volatile plant oxylipins, for example, hexanols and hexanols, are the mostly potent constituents of the smell of the freshly mown grass. They are also released in reaction to an injury. When the grass is injured, these volatile compounds are released, and they have a lot of actions. Firstly, they can directly kill bacteria. Secondly, they can alert the conspecifics that something has injured the plant. And the third action is an example of cross-kingdom oxylipin signaling. These compounds attract parasitoid wasps that are natural enemies of caterpillars and other insects that feeds on these plants. So it is a complex defense against bacteria, against other dangers and challenges, and even against insects. I always say that volatile plant oxylipins are chemical crying or chemical tears of freshly mown grass. And when we smell this pleasant scent, we can literally hear the crying of the plants. It is a chemical crying if we can expressed this way. Unfortunately, humans have no volatile oxylipins, but if we had them, we would have some super possibilities. For example, we could smell persons with the coronavirus. We know that if we have coronavirus, we have inflammatory reactions that triggers the release of oxylipins. And if we had volatile oxylipins, for example, hexanols and hexanols, the infected person would smell like a freshly mown grass, or or likewise, it would be a good alert that our conspecific, our neighbor, is infected, and we need to make a social distance. But unfortunately, we have no possibility to smell the ill persons. But plants do smell the ill conspecifics and can prepare to the meeting with the pathogen, or to the meeting to the herbivores. Directly killing oxylipins belong not only to plants. For example, diatom algae also possess directly acting oxylipins that can kill bacteria. Diatom algae are single-celled organisms. They could look like multicellular organisms, but it is a delusive impression. They have only a common mineral shell, but all these cells are totally separated from each other, and this organism is just a single-celled alga, but it has its own pathogen, the Cordia algicida. This bacterium can kill diatom algae by dissolving its cells with its enzymes. But this diatom is a bit of a lead, and it can respond with releasing oxylipins that directly kill this bacterium. It is a magnificent example of direct oxylipin-mediated immunity in single-celled algae. But diatom oxylipins are more interesting than you think, and their functions are not restricted to immunity against Cordia algicida. Brown algae and diatom algae use oxylipins as pheromones. They possess a hydroperoxidase pathway resembling plant hydroperoxidase pathway, starting with lipoxygenase, then proceeding to hydroperoxidase, and resulting in unusual hydrocarbon oxylipins that function as pheromones, attracting male and female gametes to each another. It is already not a cry for help. This chemical signal can be rather translated like, "I like you, come here," or something similar. Fungi also use oxylipins, but in this kingdom, it is already not a cry for help. Here I will take a brief spotlight on fungal oxylipins. For example, plant pathogenic fungi use oxylipins to penetrate host tissues. And other fungi use oxylipins to coordinate their development. For example, Sclerotia conidia shift, other development processes, or Mycelium yeast shift. They are also signals for developing in a specific direction, but it is already not a cry for help, but it is still a signal, but it is still a communication. And here we can conclude that oxylipins are a kind of biochemical Esperanto, a kind of universal biochemical language spanning almost all multicellular eukaryotes, animals, plants, fungi, algae, and others. And in each kingdom, it is a kind of signal, a kind of communication. And it raises a question: what is the origin of this communication? was it inherited from a common eukaryotic ancestor, or its origin can be traced even to the earlier moments when our ancestors were still prokaryotes? Unfortunately, we cannot answer this question if we use only experimental data, because all experimental data of oxylipins regard only separate branches of a large eukaryotic tree of life. For example, we are here together with fungi. All plant world is here, and brown algae and diatoms are here, and the other eukaryotic tree of view is our knowledge gap in the terms of oxylipins. This knowledge gap spans also the earlier stages of eukaryotic evolution and the last eukaryotic common ancestor. And it seems that we don't know where oxylipins came from. How did we acquire them? But we have some pegs to hang. For example, in the group of protozoa. Please get acquainted. Dictyostelium discoideum, a cellular slime mold. In the certified condition, it is just an amoeba. But if it starves, it starts to aggregate, to form multicellular aggregate and beautiful fruiting body, fungus-like fruiting body that sporulates and disseminates these organisms further. It is an interesting multicellular-like organism, but the most interesting is the fact that if its lipoxygenase is muted by mutation, by a genetic manipulation, it cannot develop normally. If the lipoxygenase of this organism is muted, it cannot proceed to the stage of culmination and stops here at the mount stage. But unfortunately, this interesting fact is not described in any specific paper. It is only a database record. Here I have inserted a link to this database. If we proceed to cyanobacteria, we know that some bacteria, but only a small percentage, only 0.5% of all bacteria living on our planet possess lipoxygenases in their genomes, so they can use oxylipins. But we don't know what is the purpose of these oxylipins. We know that the multicellular cyanobacterium Nostoc punctiforme can release oxylipins if it is wounded, injured by sonification. But we don't know who is a recipient of this signaling. And how does cyanobacteria respond to this signal? It is one more example of multicellular bacterium that use oxylipins. It is Myxococcus xanthus. It is a bacterium that in a satisfied condition is just a soil bacterium. But if it starves, it aggregates like Dictyostelium and forms multicellular fruiting bodies that resemble yellow, beautiful yellow fungi. And we know that it can synthesize oxylipins by a lipoxygenase pathway, and these oxylipins enhance its twitching motility. But we have only one article about this. So the experimental data on oxylipin functions in bacteria are still scarce and inconclusive in terms of their role in signaling in bacteria. We also know that Pseudomonas aeruginosa, a dangerous nosocomial pathogen, use oxylipins to communicate with conspecifics and form biofilms. And the same bacterium use oxylipins to suppress host's immune response. It is a complicated oxylipin fraud. When this bacterium invades our tissues, it releases its lipoxygenase that converts our fatty acids to fatty acid hydroperoxides and further to lipoxenes. The lipoxene branch of oxylipin metabolism is overregulated, and the inflammation is dampened, the protective barrier is abolished, and this bacterium can invade our tissues, damage and destroy our cells, and so it is a very dangerous nosocomial pathogen. But it is only one bacterium, only one bacterial species. So the data on oxylipin functions in bacteria are still scarce. And we decided to perform our own research on oxylipin functions in bacteria. We performed a bioinformatic research and tried to track the oxylipin biosynthesis evolution by lipoxygenases. Lipoxygenases are conserved across all kingdoms of life. They have conserved biochemical functions. They have conserved sequences. They have conserved metal binding residues. So they are useful and convenient bioinformatic proxy for oxylipin biosynthesis. We tried to perform a simple bioinformatic research and published its results in the Biochemistry Moscow journal. Here I have inserted the link to this publication. Even the first results have convinced us that oxylipin in bacteria are tightly linked to multicellularity. Here you can see the diagram of oxylipin biosynthesis occurrence in different taxa of bacteria. And here you can see that myxobacteria, social bacteria with multicellular-like floating bodies, nostochales, cyanobacteria that could even differentiate in 2 or 3 cell types. Here, vegetative cells and heterocysts are depicted. And oscillatoreales, they cannot differentiate, but they are still multicellular. So the occurrence of lipoxygenases is very high in multicellular taxa, and it leads to the assumption that lipoxygenases are associated with multicellularity even in these bacteria. The data on protozoa show a similar picture. For example, the occurrence of oxylipin biosynthetic ability inferred by lipoxygenase is high in cellular slime molds, in water molds. Both taxa comprise multicellular-like organisms. I have already discussed Dictyostelium discoideum, and sorry, it is a mistake. Here is a water mold, that looks like a fungus. It is a micellar organism, so ormycetes are also multicellular, and they also have lipoxygenases and maybe could synthesize oxylipins. The evolutionary data provided us with much more insights into the evolution of oxylipin biosynthetic capability. At this picture, it is a phylogenetic network. I have depicted the multicellular taxa in multicellular groups as blue rounds. And here we can see one more multicellular group of bacteria. It is the genus Bechatoa. They are chemoautotrophic filamentous bacteria that get energy from oxidizing the hydrogen sulfate, and they are also multicellular. So it is the third example of link between lipoxygenases and multicellularity. We have also performed some calculations to infer the group in which lipoxygenases could originate firstly. Here we can see that both cyanobacteria and myxobacteria form two clusters in each taxonomic group. We think that it means that each pair of clusters are descendants of two isozymes of lipoxygenases in a common ancestor. But who was the first to invent lipoxygenases? Our additional calculations and additional phylogenetic trees show that cyanobacteria were the first, and then myxobacteria have borrowed lipoxygenases by horizontal gene transfer. And when we proceeded to analysis of eukaryotes, this picture of horizontal gene transfers became more complicated. Here we can see that emergences of lipoxygenases are tightly correlated with emergences of lipoxygenases in each taxonomic group. Lipoxygenases spread by horizontal gene transfers, and the moments of horizontal gene transfers were correlated with the moments of acquiring multicellularity in different lineages of eukaryotes. And here we depict our reconstruction that shows that lipoxygenases possibly originated in cyanobacteria, and the cyanobacteria were the first multicellular organisms on our planet. Then they spread to other multicellular organisms. So the chronicle of lipoxygenase evolution was tightly correlated with the chronicle of multicellularity evolution, and it raises a question: could oxylipin signaling contribute to the origins of multicellularity itself? It is an interesting question that we try to answer in our research now. And our story have a striking similarity with another group of proteins, specifically metacaspases and other programmed cell death proteins described by Nick Lane in its well-known story, Origins of Death. For example, in terms of Emiliania Huxley, that we could not explain why it has lipoxygenases, but later we knew that it is able to program cell death, and it is a really striking similarity between these pathways that needs to be considered further. But it is not all. I have discussed Pseudomonas aeruginosa that could use oxylipins to evade immune response. Our results show that it is not alone. It is in the middle of some clusters of pathogenic and symbiotic bacteria that nosocomial pathogens or plant pathogens or plant symbionts. It raises an assumption that Pseudomonas aeruginosa is not the only pathogen that use lipoxygenase as virulence factor. Here is a list of other suspected species. For example, Pentoea ananas, Burkholderia gladioli, Mycobacterium abscessus. All of them are emerging nosocomial pathogens. They cannot cause a deadly worldwide pandemic like a novel coronavirus, but they can cause a silent pandemic that affects intensive care units, surgical wards, hospitals, and other sites where there are a lot of immunocompromised patients. And we have revealed a new common trait in these pathogens. A lot of them are able to cross kingdom host jumps, to jump from a plant to a vertebrate and from a vertebrate to a plant. It is an additional phylogenetic tree that was not included in our recent publication in Biochemistry Moscow. It will be included in our next paper that is now prepared by publication. Here we can see that lipoxygenases have independently jumped by horizontal chain transfers from plant symbionts to plant pathogens, to versatile plant human pathogens, and to specific human pathogens. And we can assume that this protein, this enzyme, is a universal virulence factor, and it can enable some bacteria to expand their host range and jump from plants to humans, from vertebrates to plants, and it means that we need molecular epidemiologic surveillance for bacterial lipoxygenases. Maybe we can track dangerous nosocomial pathogens by this enzyme. And I have a hypothesis explaining how lipoxygenase provides bacteria with a possibility to infect both plants and vertebrates. This pathway is already characterized in one of papers by other authors. And the second line is our hypothesis, which could occur in plants when Pseudomonas aeruginosa or another lipoxygenase positive pathogen invades them. In both cases, the lipoxygenase converts the host's polyunsaturated fatty acids to hydroperoxides, and they are further transformed to lipoxenes or jasmonates in plants that both groups of chemicals are able to enhance the invasion and suppress their host's immunity in certain conditions. The jasmonate pathway is widely exploited by some plant pathogens, and plant symbionts. But up to date, we didn't meet any publications that plant pathogens use lipoxygenase to exploit jasmonate pathway. Our bioinformatic research may be the first evidence, but it is only a preliminary result, a preliminary hypothesis. But our project is ongoing, and I hope to publish some new papers on this problem and discuss this problem in more details and provide more evidence on this hypothesis. To finish my lecture, I could recommend to read my article in The Biologist in the last issue, and a more scientifically strict and a more detailed blog post in the Nature Microbiology community. It is a kind of text version of my lecture tonight. At this slide, I provide conclusions of our own research that the multicellular bacteria and probably cyanobacteria communicate with oxylipins long before the origin of eukaryotes, and other bacteria, eukaryotes, and other multicellular organisms borrowed this function by horizontal gene transfer and use them to communicate between the cells. But some bacteria have learned to use oxylipins for virulence, and the oxylipins are fraud signals to a host's immune systems that suppress their immune responses. Here I provide some image credits, for example, to Biochemistry Moscow, to BioRender, and my friend Anastasia deserves a specific image credit. Not all arts I have used in this presentation are mine. Some arts are provided by Anastasia for free. Anastasia is a biological artist, and it draws a very beautiful arts too. And if you would get acquainted with their arts, or maybe order an illustration for your book, for your journal, for your magazine, you can write her in Instagram or by email. And here I provide some links to communicate with me if you are interested. I post my scientific results in Twitter. You can email me, you can follow me on ResearchGate, and I am launching a Substack newsletter on my research. And if just someone will subscribe me, I will soon issue a first post. Thank you very much for attending my lecture.